the point of our study was to look at those patients that developed a unique bland cholestasis that's associated with anabolic steroid use. As these patients present similar to those that have mutations in those genes that are associated with progressive familial intrahepatic cholestasis, we then underwent a whole genome sequencing strategy looking at 93 individuals of different ethnicity to look and see if there was enrichment of those genes involved with bile acid transport, and in particular ABCB11. Patients were, whole genome sequencing was performed, and then it was analyzed using a standard battery of statistical genetic analyses software to look for enrichment of potential mutations associated with genes throughout the genome. In our study, we curated 504 genes associated with cholestasis and anabolic steroid metabolism, and then used those 504 genes to then look for variants in any gene associated in that group and compare them to those who were affected with injury versus those of the population control, in which we had identified within our 93 patients those that were non-Hispanic whites, non-Hispanic blacks, Latinx, and Asian. What we found was throughout all four different ethnic groups that there seemed to be enrichment of variants in the ABCB11 protein itself, associated predominantly with the proximal membrane containing area. We then also did a pathway analysis for those genes in which ABCB11 participates, but in those individuals that did not have a mutation, and we saw enrichment of organic cation transporters, suggesting that those people that developed this either had a mutation in the ABCB11 gene, the key protein that's involved in bile acid transport off the canalicular membrane, versus potentially those genes that regulate the expression of the ABCB11 gene and thereby lead to decreased function. We then hope that by this information will allow us to better understand how anabolic steroids might cause this injury and lead to dysfunction of the ABCB11 gene.